This is the first part of the Unit 7 notes on the cell cycle and cell division. There are quite a few words in this unit that you may not have encountered before, and so here's a list pretty much in the order in which they appear. Once again, the vocabulary words should be in red, and the things that you really are responsible for writing down are in purple. This is a picture of two cancer cells in the process of division. They're almost finished dividing, and it's a really cool picture and really neat, but it is abnormal, and we'll look at some abnormal cells later on. Cell division is important for all living things. It's used for lots of different things. First of all, single-celled organisms use cell division just to reproduce. And then, of course, as a multicellular organism grows from that first fertilized egg into an adult, it takes lots and lots of cell divisions to make that entire organism. Remember, you started off as one cell, and you now have several trillion cells, and so it takes lots of cell divisions to make that happen. And then, of course, repair and replacement of cells. When you get an injury, when you get a scrape or a cut or a bruise, cell division has to repair and replace the cells that were damaged or killed in that process. Now, cell division works differently in prokaryotes than in eukaryotes, and so we'll talk briefly about what happens in prokaryotes, but remember we're going to spend most of our time concentrating on what happens in eukaryotes. The prokaryotes reproduce by a process called binary fission, and that just means dividing in half. Basically, prokaryotes are very simple. Remember, they have only one single DNA molecule as their chromosome. It's a single circular DNA molecule. It's much smaller than the multiple chromosomes of eukaryotes. Prokaryotes are very simple. Remember, they have pretty much just the cell wall, cell membrane, their chromosome, another little piece of DNA that can be sometimes passed from one to another, ribosomes and cytoplasm, and a bunch of enzyme systems, and that's really it. No real organelles or anything like that. So it's a relatively simple process to divide when you're a prokaryote. There are three main stages of prokaryote cell division. First of all, the chromosome gets duplicated. That's going to be necessary for all cell division because we want every daughter cell to have a full complement of the DNA for that organism. So duplication of the chromosome is the first thing that happens here, and then the copies have to separate from each other. As they do that, they attach to the cell membrane on the inside, and the cell continues to grow and elongate, and as it does, it moves those copies apart, and then eventually they get far enough apart for the cell to divide into two daughter cells. It's, like I said, a relatively simple process. Here we have our prokaryote chromosome that is in the process of duplicating, okay, and they're going to start separating from each other. As the cell continues to elongate, you have movement of the copies, and eventually you have a pinching off or a separation into two separate daughter cells. Like I said, relatively simple. Here's an actual micrograph of some prokaryotic cell in the process of dividing, and you can see the separate chromosomes here and here. So not very difficult, not very hard to understand, really, really simple process. It's a different story in eukaryotes, and so we're going to look at more detail in the eukaryotes. Eukaryotes undergo a process called mitosis, and so we'll talk about that, but there is an entire cell cycle of events that occur throughout the lifetime of the cell. Remember that since eukaryotic cells are a lot larger and a lot more complex than prokaryotic cells, they have a lot more things that have to happen during the cell division process. They have a lot more genes. They store their genes on chromosomes. There are almost always multiple chromosomes within the nucleus. Those chromosomes are very large and they're complex, and they have to be copied in their entirety, again, to make sure that each daughter cell is going to end up with the exact same chromosomes as the parent cell had. The chromosomes are made of a substance called chromatin. Chromatin is DNA, a long DNA molecule, and then there are proteins associated with the DNA that help keep the chromosome structure together and also involved in controlling the activity of the genes, because you don't want all genes to be active all the time. To prepare for cell division, the chromatin has to become very compact rather than all spread out. And when it does that, it then becomes visible. The chromosomes become visible with the microscope. Here you see a cell that is getting ready to divide. You can still see that it has a nucleus there, but you can definitely see the individual chromosomes and the nucleolus within the nucleus. You can't really see that except when the cell is getting ready to divide. Close-up picture of chromosome. This is a duplicated chromosome. We call the two halves of it. Each one is identical to the other. We call them sister chromatids. And as long as they're attached together, they'll remain part of one chromosome, sister chromatids. But when they separate, when they break apart at the centromere during the distribution to the daughter cells, they'll become separate chromosomes. So here's the diagram to show you. The DNA molecule is here, is replicated or copied there. The centromere, this tiny waist-like part there, is a special structure that kind of holds them together and allows for them to be separated during cell division. Once they're split apart at the centromere, they become individual chromosomes, and each daughter cell will receive one of each chromosome. Before the eukaryotic cell begins to divide, it has to duplicate all its chromosomes. And like I just said, they're called sister chromatids joined together at that central part called the centromere. One thing you'll notice about the cell cycle unit is there are lots of words that begin with C, and it's very important to pay close attention to your C words so that you don't get them confused with each other. When the cell divides, then the sister chromatids can separate from each other. They're then called chromosomes, and they will sort into their separate daughter cells. The cell cycle is what we talk about as the entire life cycle of the cell. It's a definite sequence of events that starts from the time a cell is first formed from a dividing parent cell until its own cell division, however much later that happens to be. The length of time it takes for a cell to go through its entire life cycle is dependent upon what kind of cell it is. There are two main stages of the cell cycle. The first stage is called interphase. Inter means between, and so basically it's between the dividing phases. And during the interphase, you've got to duplicate the cell contents before cell division can occur. The first part of interphase is called the G1 or growth 1, gap 1 phase. Okay, during this phase, the cell is growing, it's increasing in size, the amount of cytoplasm and the number of organelles are increasing. It's doing the job that it has to do, but growing in the process. When it reaches a certain point, that signals the start of the S phase. The S phase stands for synthesis, and in the S phase, the chromosomes get duplicated or copied. We'll talk about the exact process of how that works in another unit. And then after the chromosomes are copied and verified to make sure that they're correctly copied, then begins the G2 phase. The G2 phase is also growth, but in the process of this growth, we're now preparing for division. So all of the molecules and organelles and so forth that need to be made to prepare for the cell to divide are being taken care of. Once all the preparations are made, it's kind of like packing up to go on a trip. Once you get everything together and everything in the car, then you're ready to leave. And once all the preparations are made for cell division, then it's time for the last part of the cell cycle, which is the mitotic phase, the M phase, and this is where cell division occurs. The M phase has two parts: mitosis, which is the division of the nucleus, and it takes care of separating and distributing the chromosomes. And then finally, the part where the cytoplasm divides is called cytokinesis. This diagram gives you an idea of about how much time, how big of a part of the cell's life cycle is spent in each of these phases. You can see the entire life cycle from the first cell all the way through growth, replication, and cell division before the daughter cells are produced is encompassed by the whole circle. So most of the time is spent in the interphase part of the cell cycle, with only a very small part spent in the mitotic phase, the M phase of the cell cycle. And we'll talk more about what happens during mitosis in the next segment of notes.